Welcome to your lesson on the scientific method. We're going to run through how to do a lab report, the different types of observations that we have, and what an inference is, okay? So the main parts of a lab report when you're writing one, your teacher in elementary school might have had you write one, but the lab reports, whether you are in elementary school, high school, university, or whether this is your job as a scientist, lab reports are always kind of set up in the exact same way, and that way is called the scientific method. We want to make sure that we are doing everything in a way that our experiment can be repeated by somebody else anywhere in the world, and they will get the same results as us. That's the basic idea. So you want to basically have, kind of like when you're baking a cake, you want to have a decent recipe that somebody else could repeat it and get the same delicious cake that you just made. So we always want to make sure we have those parts to the lab. You want to know what the purpose is. Why is that person even going to try your lab in the first place? Why would they follow your instructions? What question do you have? Are you trying to solve a specific problem? Anything like that. Like, what's the main reason why we're even bothering to do this? Then after you state your purpose, you want to give an introduction. An introduction is kind of like background information. What do we already know about the thing we're about to study in this lab? What's the important stuff that somebody needs to know how to do or needs to know about in order to make sense of the results that you're going to show them? Then the hypothesis is the thing that you're testing. What is the problem that you have and what are you trying to test? But you say it in a way that you say, because I believe that this particular thing is true, so the information you've just given in the introduction, because we know that this is true, then I predict that this is what's going to happen when I do my lab. The hypothesis is very important to start with. It's okay to get your hypothesis wrong. You want to be as smart about your hypothesis as possible, and you want to make a hypothesis based on the best information. But the hypothesis is okay to be proven wrong. That's not a problem. In fact, some scientists will actually use something called a null hypothesis, N-U-L-L. Null means nothing or the opposite of. So they might use a null hypothesis, and that's basically saying, if this is true, or this is not true, then this is what I expect to happen. And it's kind of a backwards way of doing the hypothesis. A lot of scientists will do that. So either way, you're trying to prove something. That's what the hypothesis is for. Then you go through your materials. What do you need? So how many beakers do you need? What kind of powder and how many grams? How many milliliters of that particular liquid do you need? Whatever it is. You go through your materials, and then you have a procedure. Your procedure is your step-by-step, easy, clear, very easy to follow. You can repeat it every time and get the exact same outcome. Okay, has to be a very nicely and clear procedure. Then we go to, after you've done your procedure, what did you actually see? So the observations, we'll get into more observations down below, but observations, what did you see? What kind of information did you collect? And then with your observation section, you want to make that information you collected easy to read. So maybe you'll have a written explanation of what happened. Maybe you'll have a table where you write down all your numbers so that it's easy to find the information. Maybe you graph it so that it's easy to kind of see patterns in the data and stuff like that. All of that belongs in observations. And once you've done your observations, then you talk about why it's important. What is the significance of what you just found? Why did you see that? How did you get that result? What happened that got you there? Was there anything surprising in what you saw? Was there a bit of new information that you can share with the scientific community? Like, why does this thing even matter? Sometimes when we're doing analysis in high school, we'll just ask you to answer direct questions from what you saw. In other words, what's the importance of what you observed? So if you saw, let's say you added one liquid to another and all of a sudden there were bubbles. The observation might be, I saw bubbles. The analysis would be, a gas was produced. That explains why those bubbles were suddenly there. So you can say there was a chemical reaction, there was gas produced, and that would be your analysis. And then the discussion part just talks about why that matters. And you could also, in your discussion, you could talk about maybe any limits to your particular things. Let's say you're trying to go and research a cancer therapy, and you've gone through and you've done your procedure and you've written that all out and you've gone through and you've tested the people, you've given certain drugs to some people and you've given a fake drug to other people. This is what you observed. You observed that the people in the drug group were doing better with the cancer. They were beating cancer. The group that didn't have the drug, it was same, nothing changed for them. That's your observation. Your analysis says the drug must be working. Your discussion says, here's why our stuff is important. We believe that the next person needs to do even more research, what would they do in their research? Well, you can give some suggestions in the discussion section and say, in the next test, we would have used, and you can say maybe a different dose, or maybe you would have had 100 people in your test rather than only 50 people in the test. Maybe you would have done some things differently. So you can say what could have gone wrong, and you can say what people should do next time in the discussion section. So this is really looking at your results and saying why does it matter? And then your conclusion always goes back to your original hypothesis. Did you get it right? Did what you think would happen? Or did you see something else? And if you saw something else, then you explain it in your conclusion and in the discussion section. Okay? So that's the overall idea of how we set up a lab. Have your purpose, give some background information in the introduction, tell us what you think is going to happen based on what you already know, lay out everything you're going to need for somebody to repeat your procedure, write out your observations either in charts or tables or in written information, analyze and discuss your data, and then come to your conclusion. Did you see what you thought you were going to see? If you didn't, why? So that's the overall idea of the scientific method. Now let's talk directly about observations themselves. Direct observations, you're going to say, this is something that I actually saw myself. So I was able to look at the table and I saw it was gray. I was able to grab a ruler and measure the length and width of an object. You directly saw these things yourself. Maybe you were looking for, I don't know, maybe you were trying to detect radiation though. Now radiation you can't see with your eyeballs. You can't feel it on your skin necessarily unless it's something like heat or something like that. You can feel heat. But maybe you didn't directly detect it yourself, so you used a special, some kind of an instrument like something like a Geiger counter. A whole bunch of different Geiger counters here. And basically what happens is this little black part here is a detector, and the detector can pick up radiation. And so you walk around with this little box, and then if you're near radiation, you hear that tick, 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 tick. And as you get closer and closer to the stronger radiation, it ticks more. So you can hear it, and you can see it on the scale to see how bad it is. So how many times the radiation is slamming into this particular device every second. So how many counts per second, okay? You guys have probably seen those in movies. So something that's indirect, you're probably going to need to use some kind of a special detector in order to pick that up because you can't detect it yourself. An indirect observation could also be something like when you have the wind blow past you. Let's say you're standing outside in the yard and you're looking up at the trees and the wind blows past. You can feel the wind against your skin, but you can't see it. What you can see, though, is the wind wiggling the leaves on the tree. And when you look at the wind moving the leaves on the tree, that's an indirect observation. You can say, I can see the leaves moving, so indirectly, I know that there is wind. I can observe the wind by what it does, even though I can't see the wind itself. So that could be another example of an indirect observation. Test questions will probably ask you to define the difference between them and give me an example of each. So make sure that you're able to do that. Then we get into qualitative and quantitative. Qualitative observation is one that you don't use any measurements for. So if you touch something and it feels smooth or rough or slimy, you're judging the texture of that thing. That would be qualitative. You're not putting a number to it. You know, looking at a dog and deciding whether it's a Chihuahua or a Great Dane, that's qualitative observation. Color is a qualitative observation, unless you get a special machine that can give you the exact wavelength of the light that gives you the color. Quantitative, on the other hand, always has a measurement to it, and we always use some kind of a unit. So if you're measuring the mass, you can measure it in kgs, or you can measure it in pounds or grams or whatever. Distances can be measured quantitatively in kilometers or meters or feet or inches. Time can be measured in seconds, hours, days, years, milliseconds. You can even go smaller, right? So quantitative observations always have a number, and then qualitative never have a number when you describe something without having a number. Please memorize these two, definitely on your test. An inference is basically what you conclude. What's your final judgment on what you just saw? So a judgment based on your observations. You don't directly necessarily see it. You're saying, because of all these other things that I've seen, I'm going to decide why this thing is happening. So if I picked up a stapler right now and I dropped it, you would see the stapler fall. That's your direct observation. You saw it with your own eyes. But why did it fall? Well, the why is the inference. We would say right now, with our current understanding, we'd say, oh, the force of gravity pulled it down. We inferred that. We didn't know that for sure. So here's our little setup for the lab. we've got in this container over here, we've got something called soluble starch. It's this white powder that I've got sitting here. And all I did was take that white powder and I put it into some water. And you can see that here. So if we were going to make some observations of this liquid right here, we might say maybe it's got a slight translucent color to it. It's not quite perfectly transparent. There's a little bit of maybe whitishness to it, but it's pretty almost clear. It's got the same viscosity as water. The viscosity means how easy it flows. Same viscosity as water because it is actually water. It's just this white powder floating around in the water. Okay, so translucent. And then we have this stuff. It's called iodine. Let me show you what iodine looks like when it's out of the bottle. So you can see the iodine itself has this yellowy, kind of a golden color to it. Iodine is actually a powder. This has also just been put into water, okay? So now we're going to think about what our inference is going to be. We should know that in the presence of starch, this iodine will turn like a bluey, blacky color. So let's just prove that. I'm going to take the starch that I've got here, okay? I know for sure it's starch because I took it out of the bin myself, and I took it out with this little scoopula thing, okay? And I'm just going to take the starch and I'm going to add some iodine to it. Do you see, even though it's yellow being dropped, you can see very much so you get this really, really dark color, this blacky, purpley color that'll actually... So that tells us that there is definitely starch in this particular thing. It's not some other type of powder. It has to be starch because of that iodine test, okay? So then I can take two other liquids that I'm not sure about. All right. So I take these two liquids, and I'm not sure which one is starch, but I want to do a quick little test to see which one. All right, so we can start off and we say, what are our observations when we start? Liquid on the left here is a little bit whiter in color, but it's still kind of a translucent liquid. And this one is a little bit maybe browner in color, but they're so very, very close. They both look kind of the same. And I want to figure out which one has starch in it. So I already know the relationship between iodine and starch. I should see that blackish color, okay? So I take the iodine and I'll add it to liquid number 2 over here on the right. Okay. Did you see any color change? You tell me. Did the iodine change color? The iodine was always yellow, so not really. I mean, it... Yeah, so this is what it looked like before, but that's just because I had concentrated iodine here, very concentrated iodine, whereas here I've just got less concentrated. So let's just add water to this one and see if I can get it to be a little bit more of that color. Okay. So now I've got the original iodine that I had. I just added water to it. So now I've got the translucent liquid with the iodine in it, and I've got the iodine just pure itself. You can see it hasn't really changed color. The iodine is still very much so yellow, okay? But when I go to this other one here, this other transparent liquid, if I add iodine to it, if it's not starch, it should do the same thing as that one down there. It should still say yellow. If it is starch, I'll see a dramatic difference. Okay? And you can see right away, okay, for sure, all that color that's in there, for sure we're talking about starch being present, okay? Massive difference in the results. Let me get rid of that because that's just iodine. So a massive difference in those two results. The test tube on the left, we can infer, this is why we're doing this and showing what an inference is. We can infer the test tube on the left does not have any starch in it because the iodine did not change color. And we can infer that the test tube on the right did have starch in it because it did change color, okay? I just wanted to give you that example of what an inference is. Before we put in the iodine, we had a white liquid. It was translucent. After we put in the iodine, it turned purpley black. So for sure starch was present. In the other one, it's kind of a transparency clear liquid as well. It stayed pale yellow, so there's no starch. So the inference is, the conclusion drawn, is starch must have been present. So please do a good job of memorizing these 5 terms and know what we're talking about. And as we're going through different types of labs, make sure that you're able to in your mind using the terms qualitative and quantitative when you're looking at stuff, and know what the word inference means. So now let's chat about scientific method is in terms of variables and organizing your data. Okay, what's a variable? Well, a variable is anything that you can change during an experiment. So even if you were to change the length of time that the experiment was going for, that's a variable. Time would have been your variable. It's the thing that you changed. Let's say you, in one experiment, you had it at, maybe you had your water sitting at room temperature, but in the next part of the experiment, you had your water boiling. Well, temperature then is your variable that you changed for your experiment, okay? So anything that you choose to change. Most experiments, because of the scientific method, we actually don't want to change the variables a whole lot. We want to keep them what we call constant. Constant means keeping them the same. And usually you're only looking at two things at a time that you're going to allow to change. Those two that you will allow to change are called the dependent and the independent variables. This is what you guys have been doing in your elementary school graphing. You've been using the independent variable and the dependent variable, and you've been graphing them in charts, right? The independent variable is the one that you as the scientist are going to choose, and you control how much it's going to change. And the idea is that the independent variable that you're controlling goes on that x-axis, but the main thing is that the independent variable that you are controlling is going to change the other one, the dependent variable. It's called dependent because how much the second thing changes depends on how much you as the scientist changed the first thing. So this one's allowed to change freely in response to whatever you did with the independent variable. So your independent variable always gets graphed along the x-axis, along the horizontal axis, and your dependent variable always gets graphed along the side. And you'll find that anything that you're timing, time is always an independent variable, because how long you let it run for is what you're controlling, how long the time goes for your experiment, and then how the other thing you're looking at reacts to that time is depends on that time. And again, that's always placed on the y-axis. So let's do a couple little examples here. This again is test type of questions. You want to make sure that you have a good understanding for this. And not even just test questions, but just in general, if you don't know how to use the independent and dependent variable, you're kind of stuck for both math and science labs in high school. Okay, so make sure you really get a good solid grasp on this stuff now. So for example, let's say we're throwing a ball, and so we're looking at how high the ball goes over a certain amount of time. So you throw it up and you are timing, you click on your timer so then you can actually see how far it goes in a specific time. So imagine it's going straight up, straight down. Your independent variable, the thing that you're controlling, is the time. You're looking at how long are you letting it go for, or how long are you actually clicking the timer for. The dependent variable would be the height, because how high the ball goes depends on what time you gave it. So in other words, if you throw up the ball and it's still in the air when you turn off your timer, well, you controlled the time. You turned it on and turned it off. And when you're doing your little video of the ball flying, you're only going to get enough data to get you to the end of whenever you turned off that camera, right? So the height would depend on when you shut the camera off, when you shut the video off. Even though the ball will drop after that, it's how long were you running it for? How long were you looking at it that matters, okay? So height would depend on the time. The time doesn't depend on the height. Another example, how long you spend doing homework compared to the mark that you get on the test. The time that you spent, the hours spent would be your independent variable, the thing that you controlled how long you went for. Your final mark should depend on how long you studied for. How long you studied for shouldn't depend on the final mark. So even if you put it into a sentence like that and say, final mark depends on time studied, and then reverse it and say, hours spent depends on the final mark, you can see one of them makes sense logically and the other one doesn't, and that might help you to figure out which one's dependent and which one's independent. Obviously, the dependent always depends on the independent. Let's say the width of your thumb compared to how often you win at Thumb Wars. Well, then you'd be looking at how often you win depends on how wide your thumb is. How wide your thumb is does not depend on how good you are at Thumb Wars. You can have a wide thumb and still be terrible at Thumb Wars. In fact, we've done studies in the past here with grade 9s when we were actually allowed to touch each other pre-COVID, and we would do a thumb war thing, and you'd actually find out that the width of your thumb really doesn't help you in a thumb war at all. It's usually actually more how competitive you are, how determined you are to win. That's really what helps you win a thumb wars. Even the length of your thumb is a little bit of an advantage, but that's about it. So anyway, we've done some experiments with that before. So that's dependent and independent. Let's take a look now at the controlled variables. So again, we call these constants, controlled variables constants. It just basically means you're trying to keep something the same for the entire experiment, or at least the entire portion of the experiment. So let's say I'm trying to see how fast a speedboat goes, and I want to test its engine. I want to see an acceleration test. This is a fancy way of saying you speed up the boat as fast as you possibly can to see how fast it can go in a certain period of time. So the speed measurements, let's say they're taking 10 seconds for 1 minute. So let's say at time 0 we do the speed, at 10 seconds, 20 seconds, 30 seconds, 40 seconds, so on, all the way up to 60 seconds, we're measuring the speed, okay? And we're going to see then how much it can accelerate at each of those time intervals. So at 10 seconds, how fast is it going? At 30 seconds, how fast is it going? At 60 seconds, how fast is it going? So how fast it's going depends on what time you looked at. The time doesn't depend on how fast it's going. Controlled variables, though, those are the dependent and independent. Controlled variables would be things that you never let change. So the time changed and the acceleration changed, but you want to keep everything else the same so that you know that your test is working. So let's say you're out on the water somewhere. You want to make sure that your wind speed's the same. Are you heading into the wind or do you have a tailwind? Where did you actually put the throttle? Did you always put it up to 3/4 or was it on full the entire time? How heavy was your boat? Were there a lot of people in it? Was it empty? What kind of fuel were you using? Using the best possible fuel or using the junky stuff? All of that stuff matters. So if you want to be able to test between different types of boats, you want to keep as many of these things the same as possible so that you can see, is speedboat A really faster than speedboat B, or did you have a better fuel and that's why it went faster? Or did you have less weight in the boat and that's why it went faster? Or did you have the throttle on full and that's why it went faster? So keep everything the same as much as possible so that all you're actually comparing then is the acceleration between the different boats. Okay? So that's what a controlled variable is, keeping things constant or the same for the entire experiment so that you know it's not winning because of these things, it's winning because it just happens to be a better engine. So we can also have something called an alternative experiment. This is when you have two groups, they get set up with different independent variables. So you're actually not looking at the same thing. So this would be like the cancer example I gave earlier. Your control group, let's say, has totally normal conditions. Everything's kept exactly the same. Okay. Then the test group, you're going to just change just the independent variable, and you want it to just be testing and seeing how it changes, hopefully just one thing. So what you're doing then is everything else that the control group has is identical to your test group except for one thing. So let me give you a real life example. Let's say, again, we're going back to the testing a drug kind of a thing. Let's say that your test group, the one you're testing, they get the drug and you see how long their symptoms last for. So let's say somebody's got allergies. Let's say it's an allergy drug. So they receive the drug and they look to see, do they still have their symptoms for a specific period of time? So let's say they look at them over 4 to 6 hours or something like that, as long as the pill should be working for. Do they still have their symptoms, or does their, they stop coughing? Do they stop having their nose run? Stuff like that. The control group doesn't get the drug, but they have the same issue. They have the same allergies, and we look to see, did the drug help anybody out more than just the people who never even got it? So exact same symptoms, did the drug do anything? That's the difference. That's called an alternative experiment. What would be the dependent variable in this case? Well, that would be how long the person's sick for. How long do the symptoms last? Were they able to clear up the symptoms? So I'm looking between the test group and the control group. The only thing that I changed was who has the drug. So that was my independent variable. It's the thing that I controlled. And then the dependent variable was, did it help them? Did it relieve symptoms? Was there a shorter length of the illness and whatnot? Let's just finish up now and we'll talk about organizing your observations that you're going to be doing. Tables are the fastest way to help you record stuff. Take a look at this information here on the right. This is called prose. Prose is when you just write things out. Now, this prose doesn't even have a quarter of the data that's in this table. This table is much easier to read. The table title is very informative. It tells us, okay, look, we're comparing the mass, the diameter, the density, and the gravity, great, of celestial objects. What are celestial objects? Those are things that are up in space. So we've got Mercury, we've got Venus, the Earth, the Moon, Mars, and so on. So we have 5 celestial objects, and we've got everything nicely laid out. It's easy to compare. And you can see that's way better than trying to compare it to things being written out here. It's just kind of a garbled mess. And over here on the right in the prose, in the written way, we don't even have half the information that's over here. They must have a nice title on them. Always have an informative title that tells you what the data is about. Then we're going to get into graphs. Graphing is a visual way to compare stuff. So if I want to compare the height over time, I can do that very nicely with a graph. Keep in mind, because I have numbers, I can only do quantitative data this way. I'm not going to put necessarily colors on a scale or the way something textures and feels. I can use a table to write how things feel. And I can describe something feels slimy or smooth or rough, but I'm not going to necessarily put that on a graph. I can, but not necessarily, because I want to use numbers on a graph. So here I got a nice descriptive title. You learned this back in elementary school. You need a good title. You need to have the axes themselves. This is the y-axis and the x-axis. Every box has to be the exact same amount. So if you decide that this first box here is going to be worth 1, then this second box has to be 1 as well. Has to be 1, has to be 1. You can't just say 1 and then all of a sudden put a 10 over here, because then this box is only worth 1 and then this box is worth 9. You can't do that. So you want to make sure that every single time you're doing this, that your scale is always consistent, okay? Whether it's the x-axis or the y-axis. That's really, really, really important. And then your data points just fit. Once you've got your scales, you put your data points to fit the data. Now, a lot of you guys are thinking, of course, that's how you do that. But there are always some people that don't know that, and they do it incorrectly. So I want to make sure that you know. Super important. Scales have to be consistent. Always make sure you label your axes. When I test you guys, I'm going to be looking for that. You have to have the actual label, so time, and you have to have the unit. Was it in hours? Was it in days? Was it in years? Nope, it was in seconds. So I need to know that. Same thing over here. You need to have a label for here. Height in centimeters. You need to have your graph large enough, easy to read. You don't want to have all of this data squished into this little tiny area. You want to use the space so that it's easy. You want to be very accurate with your points when you plot them. Don't just sort of fudge it and guess it. You want to be very accurate. And then we don't just want the dots. We want to show the pattern that we see within that data. So we would either draw a line, which would be a straight line with your ruler, or a curve of best fit. And in your grade 9 math class, if you haven't taken math already, you will be going over exactly how to do that. And again, we're going to use that to show patterns, but we're also going to use it to help us predict. And this brings us to our last point here. You can see I've got where the circles are. These are my actual data points. So I know for sure that at 4 seconds in, the ball was at 45 centimeters high. And I know that for sure 7 seconds in, it was at 30 centimeters high. But I don't know for sure where it was here at 1.5 seconds. I don't have the actual data point for it. I never tested it. I didn't look at it at 1.5 seconds. But let's say I want to know. Well, once I've done my graph and I've plotted my points and I've drawn the curve of best fit, then I've got information in between my data points that I can make a good guess at. How did it get from 1 second to 2 seconds? It must have been somewhere around, maybe let's say 23 at about 1.5. So I go up 1.5 to the point and I go across and I say, yeah, it was probably maybe 23, maybe 24 centimeters high at 1.5 seconds. Okay. The idea of going in between the data points that you have is called interpolation. Inter means between. So that's when you're predicting information between the points that you actually know. So you're within your data range. Inter, between. Okay. Let's say though, see over here at 7 seconds in, that's the last time that I actually took a picture of the ball and knew exactly what height it was at. I didn't actually see the ball fall. But I want to guess and I want to say, well, I know where it was at 7 seconds. How long did it take the ball to fall? So what I do is with my curve, I draw my curve up and around, and then when I get to this point that I have, I just try to follow the pattern. So whatever my hand was doing when it started to curve down, I just keep drawing that line. And as I keep drawing that line, what I'm doing is predicting where the ball would be later, beyond the time that I actually was watching it for. So at 7 seconds, for sure it was at 30 centimeters. I can read that right off my graph. But at 8 seconds, I can guess it was just below 20. 20 would be right there. So maybe let's say 18 or 19 centimeters. I'd have to guess, but I try to be as accurate as possible. For sure, it's not 20. 20 is too high, so I'd have to say 18 or 19 at 8 seconds. The idea of guessing beyond your data when you don't have information, but you're trying to use the pattern, is called extrapolation. Extra means beyond. So trying to pick a point beyond your data. And when you do that, you are extrapolating. So now what I'd like you to do is to do this assignment right here, working with tables and graphs. And what you're going to do is, using this data, you're going to determine, first of all, which one do you think is the independent variable. You have to choose that first because your independent variable is going to go along the bottom and your dependent variable has to be written along the side. So if you don't know which one's which, you're not going to be able to graph it. So basically you have to kind of picture what's going on here. You got a rubber band. It says here the weights added to the rubber band will cause the rubber band to stretch. So let's say you got a heavy weight attached to a rubber band, and now if you can picture that weight is, you're holding onto the band at one end and you've got the weight on the other end and it's stretching that down towards the ground. How much weight you put on it will change. That's what you are controlling. And then the length of the rubber band is going to change depending on how much weight. So we would say the length of the rubber band depends on the weight that you chose. The weight the weight of the actual object you're shoving on it doesn't change ever. The 25 gram weight will always be 25 grams. It doesn't depend on how long your rubber band is, but the rubber band stretching does depend on the weight. So we would say that the length is the dependent variable, and then the weight is the independent variable, because this is the thing that you chose, this is the one you're changing, and you're seeing what it does to the length. So once you know that, that means that the weight as the independent variable then goes along the bottom, and then the length numbers will be along the side. Then you're going to graph the data. Make sure again that you are doing nice consistent scales. So if you decide, let's say here, we're going from 25 all the way up to 200 for our weight. So you count the number of boxes. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26 boxes. Okay? You have to decide what is every box going to be worth so I can make sure that 200 fits on my scale. So I would say if you went up by tens along the bottom, so 10, 20, 30, 40, 50, and so on, then you would be somewhere around here when you got to the 200, which is great because you've used a lot of your space, but you leave just a little extra space so you can guess beyond your data, so you can extrapolate beyond your data. So maybe going up by tens along the bottom, okay? So that means that when I go to graph 25, I'm going to have 10, 20, so 25 will be here, and then it'll go up as far as I need it to go to get to my length. So do the same thing for length. Make sure that you're very consistent. If you're only going up to 10 and a bit, let's say 11, that would be the next one beyond that, then maybe you could go up by halves. So every block here would be worth half. So every 2 blocks is worth 1. And then you're just consistent all the way up. So that way you've got a nice, good, consistent scale. You plot your dots, and then you're going to draw either a line or a curve of best fit, whichever one you think looks better. And so you're going to be looking at all your dots. You're going to say, that's kind of in a line. I'm going to do a nice straight line with a ruler. Or no, there's a very obvious curve in the data, so I'm going to do a curve. Okay. Then using that curve, you're going to either extrapolate or interpolate. It depends what the question is asking you for. Extrapolate, remember that that's beyond the data. So beyond 200 grams. So if you were to stretch to 250 grams, what would be the length that you can predict for that rubber band if you happen to have a 250 weight. Okay, so then you give that there. This question here, you don't have to actually draw a graph. You're just looking at the data, trying to decide which one's dependent, and then trying to predict and say, would you have to interpolate or extrapolate to find the answer to this question? So that, my friends, is the end of this portion of our lesson.